So this is the exception to the dominance. The first exception is the incomplete dominance. Second we are going to learn is the codominance. So incomplete dominance is the phenomenon where none of the contrasting allele is dominant. An expression of the trait is an intermediate, or you can say blending, intermediate or a fine mixture of the expression of the two factors. So NCERT gives you only one example, Snapdragon antirrhinomagus. If you need another example, it is 4 o'clock plant which comes from Peru. I've told you it's also called as the miracle plant, also called as the Mirabilis jalapa. So in this plant also you can see the same feature, that there will be incomplete dominance. So a cross between varieties with red and white flower produce all the pink flower, and on selfing the phenotypic ratio came out to be 1:2:1. Here pink have heterozygous phenotype. This phenotypic genotypic ratios are same in the case of the incomplete dominance. So how it happened exactly is the concept which is given in NCERT, and I have written in the notes, and I will try to explain from the notes. Otherwise you can open the page number 76 of NCERT and you can learn from the last paragraph of the NCERT from there. So what exactly happened is the reason behind the incomplete dominance. So we all know what is mutation. Change in the DNA is called as the mutation. Change in the DNA means change in the polynucleotide, or you can say even a change in the smallest portion of polynucleotide can also be called as nucleotide or a base. You remember bases we learned in 11th class, right? A, G, T, C. Okay. So let's imagine that there was an original allele. Allele is what? It's the DNA again, which is located at a particular location in the chromosome, a fixed chromosome, a fixed position called as locus. So an original allele is there. It underwent a mutation. Mutation can happen due to any reason. It's a very long topic. We'll be learning in this 12th class only, but not now. So mutation occurs in the nucleotide, and maybe one nucleotide, 2 or 3 nucleotides, they underwent a change. And once the allele is changed, there will be change in its expression. So there will be an altered expression of the allele. Now what will happen? If there is a mutation, 3 things are possible. Those 3 things I have mentioned here also. It is possible a mutation has occurred, but there is no change in the expression. So such type of mutation is called as the same sense mutation. What is same sense mutation? When there is a mutation in the DNA, but it does not bring any change, so the meaning remains same. That's why it is called as same sense mutation. Second thing possible is there is a mutation in the DNA, and now it causes the formation of a different type of information. That means it is called as the missense mutation. What is missense mutation? That a change occurred in DNA, and now the type of the feature or phenotype expression has been completely changed. In same sense, phenotype remains same. In missense, something new occurs. Now it is nonsense mutation. Sometimes a mutation in the DNA will cause formation of no phenotype. So because there is nothing formed, it is called as the nonsense mutation. Now same thing are explained in detail, but not completely in the NCERT. So NCERT has given this much at least. So original allele undergoes mutation, and now there is a change in the expression. And as the expression means the phenotype. So as the expression is going to change, it is possible. Maybe the mutation will cause. Okay. We all know DNA forms mRNA, mRNA forms the protein. This was the central dogma to the 11th O section students I explained last year. So what exactly happens is DNA change will cause a change in the RNA, or to be more precise, mRNA, messenger RNA, and this change will cause a change in the protein. Now proteins, when I say here we are talking about enzymes, maximum enzymes are proteins. So if the enzyme is changed, it is possible. A changed enzyme could be defective enzyme and it is no more functional. Or it is possible the change in the DNA is so disastrous or so much different that there is no enzyme formed. So here an enzyme is formed, but here a protein is formed, but it is defective in nature, it does not function. Or nothing is formed, no protein or enzyme is formed, or there is a functional enzyme formed, and it is possible it can be same functional enzyme or something new. That means it is causing something change in the expression. Let me repeat again. There is an original DNA, it undergoes mutation. This mutation depends upon how much drastic is this mutation. It can have a huge impact or it may have no impact. So it all depends upon chance factor. So a change in the DNA occur and then the expression is going to change. If there is now, because the DNA has been changed, mRNA will be changed. As the mRNA is changed, protein would be changed. Now a change could be of various type. If the DNA has been changed too much, it is possible no enzyme would be formed. It is possible a protein will be formed, but it will be a non-functional protein, so it won't act like enzyme. Or it is possible it may cause a formation of a different type. It's a functional enzyme, but it has been a changed enzyme. Now if the enzyme is changed, it may cause formation of completely different type of expression. So maybe the features are going to change. So anything is possible. This is given in the NCERT, and this is the reason why incomplete dominance has come into existence. We'll be learning this is the reason also for codominance also. So I hope this much is clear.